okay, the nerve message, what's actually taking place here. So when you think about the neuron, and you have to consider it is a cell, and it has this cell membrane, this plasma membrane as such. You have these ions, and it's the ions we're talking about are sodium and potassium. That's the two we focus in on and such. But they both have a positive charge, just that the potassium has a much bigger positive charge than the sodium. So the sodium is always on the outside of the cell membrane, and the potassium is just right on the inside of the cell membrane, but it has a big positive charge. And so it attracts or accumulates all of these negative ions. So it makes the inside of the membrane look or appear more negative than the outside where the sodium's found. So it has this difference in charge, this polarity and such. So the outside is more positive than the inside. So, and they measure that in what's called a resting potential. It's a potential. Think of it like water behind a dam. It has potential to do work. So you have all this sodium on one side, all this potassium on the other side, and then there's a potential to change, a potential to do work, if you will. And this is measured in volts, millionths of volts, millivolts and such. So when they cross, when the ions cross, that is how a electrical charge moves across or moves down a membrane. So they measure this, and this value, which, remember it, stick it in your pocket, what's it going to do for you? But it's negative 65 millivolts. Okay, that's the resting potential. It's a negative because it has this ability to do or to change, and also now that's due to the sodium being on the outside, the potassium being on the inside, and then all of the negatively charged ions in the cytoplasm and such. So how are these ions kept on opposite sides? By a special protein called the sodium potassium pump. This pump runs using ATP continuously, and it moves them in a special ratio. It will pump 3 sodiums out at the same time it'll pump 2 potassiums in. So we say that it's a 3-to-2 ratio. I'm doing that right there. That helps to maintain, or that does maintain, this imbalance. Sodium on the outside, potassium on the inside. It's also needed to, when this balance is disrupted, to reestablish that, or to reset that charge and such. So it is actively, so it uses ATP, actively pumping these ions against their gradient. So it's moving both of them, if you will, uphill. Now this graph right here, this shows you what the resting potential should be, about negative 65. And then that pink line, or fuchsia, whatever color that is, that's the threshold. If a charge distribution stays below that, then an action potential won't be generated. Once you go past that threshold, then it's an all or none, if you will. As you go from the negatives to the positive, that's the sodium running into the cell. Then you get to the apex, and then it drops back down into the negative. That's the potassium rushing out of the cell. And then that little bottom dip down there, that is the sodium and the potassium being put back, the trigger being reset, if you will, on that action potential. That's known as a refractory period. So this action potential is just a temporary reversal of the charges. And it's due to the sodium gate, which is a protein, and the potassium gate, which is also a protein, opening to their respective ions, allowing those ions to cross very quickly. Basically, they change positions, and then that is what's happening. And also, when this happens, when the sodium comes in, that is the nerve impulse, and then when it gets reset, that's it. And then when it gets put back, when the sodium gets pumped out and the potassium gets pumped back in, it cannot be re-stimulated at that point in time. So, but this is what's happening. It's just these two ions changing places. So the sodium goes first, and then once a big chunk of it has gone in and the charge gets up to a very high positive charge, that then causes the potassium gate to open. So, and then the potassium goes from the inside to the outside, and that reestablishes or resets the positives and the negatives where they're supposed to be. So when the sodium runs in, we say that the membrane has become depolarized, and when the potassium runs out, we say that it has been repolarized, and then it gets reset, this refractory period. So that's, in a nutshell, what happens. Okay, so now that sodium potassium pump is constantly running, and it resets this charge fairly quickly. So now on a membrane, where the action potential starts, it moves out. It can't go back because of that refractory period. So once you have it generated in one area, it moves out in all directions. And then the next section can't be, it depolarizes and it can't be re-stimulated. And it causes that action potential to keep moving away from the original stimulus or starting point, so that the message, the action potential, propagates down the membrane, propagates along the membrane, and all. So, but that refractory period is where that membrane cannot be stimulated a second time. So, looking at it again, third time here, when it's at rest, sodium's on the outside, potassium's on the inside. The outside is more positive than the inside. And then you have a stimulus, whatever that is, and the sodium very quickly moves in. That causes the inside then to be more positive than the outside. That's the depolarization. Once that's reached a certain positive charge, then the potassium gates open and the potassium goes from the inside to the outside. That is repolarizing, repolarizing the membrane. But it can't fire. Okay, it just doesn't fire again. It'll only generate an action potential or fire when sodium's on the outside. So then that has to be reset. So the sodium gets pumped from the inside to the outside. The potassium gets pumped from the outside to the inside in a 3-to-2 ratio. And it resets or restores the original polarity of the membrane. And then boom, you have it. So there you go.